The previous video introduced the basic knowledge of fluorescence and antigens. The theme of this session is multicolor panel design. First, different fluorochromes have spectral overlap. Therefore, when choosing fluorochromes, try to use combinations with minimal spectral overlap to reduce the complexity of data analysis. For example, FITC, PE, CY7, and APC have no spectral overlap. The FITC channel is also known as a non-interference channel, meaning that other channels will hardly interfere with this channel. When designing panels, this channel can be used for important markers. It should be noted that FITC is sensitive to low pH. Avoid using FITC in low pH environments. PE/cyanin-7 and APC/cyanin-7 conjugated fluorophores have poor light stability and are not resistant to fixation. Use them cautiously in experiments that require fixation. Cyanin-5 nonspecifically binds to Fc receptors on the cell surface, increasing false positives. Therefore, avoid using cyanin-5 fluorochromes in cells with high Fc receptor expression, such as macrophages, NK cells, and monocytes. Secondly, different antigens have different expression levels, and different fluorochromes have different brightness levels. When designing a panel, note that strongly expressed antigens can be paired with weak fluorochromes or strong fluorochromes. Weakly expressed antigens must be paired with strong fluorochromes. For example, the strongly expressed antigen CD3 can be clearly separated whether using weak or strong fluorochromes. CD25 is a weakly expressed antigen. Using PE, a strong fluorochrome, the separation is clear, but with per-CP, a weak fluorochrome, the positive and negative populations cannot be separated. Therefore, weakly expressed antigens must be paired with strong fluorochromes. If the expression level of the antigen is unclear, the most important antigen in the panel should be paired with the strongest fluorochrome. Lastly, let's look at the relationships between markers: mutually exclusive, progeny and parent, and co-expression. When designing panels, mutually exclusive antigens allow spectral spillover. Generally, allow progeny to spill over into parents, but avoid parents spilling over into progeny. For co-expressed, low-expressed antigens, avoid spectral spillover. The goal of panel design is to minimize the complexity of analysis. While it is often not possible to meet all the requirements, we should try to minimize the complexity of data analysis through these principles. Therefore, there is a saying, there is no best, only better. Here is a specific example of panel design. Detecting mouse spleen Treg. Initially, the fluorochrome scheme was CD4 with PE, CD25 with per-CP/cyanin 5.5, and FOXP3 with FITC. As shown in the figure, CD4 separation is clear, but the CD25 and FOXP3 double positive population is not very clear. This is because both CD25 and FOXP3 are weakly expressed antigens, and per-CP/cyanin 5.5 and FITC are not very bright fluorochromes. By changing CD25 to APC, a strong fluorochrome, and FOXP3 to PE, a strong fluorochrome, the CD25 and FOXP3 double positive population became much clearer. Today's flow cytometry lesson ends here. In the next session, we will discuss how to solve panel design problems. Follow us on YouTube by scanning the QR code or find us on TikTok by searching eLab Science. Let's unlock more flow cytometry knowledge together.